Hello there. My name's Dr Steve Jacques, and I am an anatomist who works at the University of Leicester Medical School. I'm particularly interested in the structure and function of the nervous system, and so for the next half an hour or so, I want to talk to you about some of the methods that neuroscientists have used over the years in order to work out how the nervous system works. Now, some of you might have been intrigued by these images at the bottom. This one bottom left is an MRI walkthrough of the human brain, and we can see very nicely the soft tissue detail shown by this incredibly powerful imaging technique. On the right, we can see a thick section through the rat brain using a technique called rainbow, where all the different types of cells have been labelled in different colours to show us where they are in the brain. So let's have a little bit more detail on these different methods. Now, for neuroanatomists and neuroscientists in general, we have a very fundamental problem. The brain is this relatively amorphous blob of tissue. When you look at the brain, you can infer very, very little about its function just by looking at it. However, we do know, and if any of you have actually looked at any books on neuroscience, we do know that within this brain there is a very, very intricate wiring pattern. Okay, it's a huge amount of complex connections within the nervous system. And the problem for neuroanatomists is, how do we go from this blob of tissue to this complex wiring diagram? Before we embark on that journey, let's just do some very, very simple revision. As you should know, the fundamental unit of the nervous system is the neuron. And here in green is a typical neuron that's been filled with a fluorescent dye. You can see that the neuron, like all cells, has a cell body containing the nucleus, and it receives inputs from its dendrites. So all these are dendrites which receive inputs from other neurons, and it is on these that the synapses are found. The neuron adds all these influences together and essentially makes a decision about whether to fire or not. And that decision about whether to fire or not is sent along the axon that we can see. Okay. So at the most fundamental level, a neuron is a decision-making device. Now, neurons don't exist in isolation. They exist in great numbers, in huge populations within the nervous system. And if we look at a cross-section through the spinal cord, as it is in this case on the bottom left, we see a whole number of neuronal cell bodies here. And we can really see the density of the axons and dendrites within the neural tissue. Okay, it's incredibly crowded, a huge number of connections, synapses, dendrites, and axons in here. Not to mention the fact that we have a whole load of glial cells, which are smaller cells filling the gaps between the neurons, doing a whole lot of very interesting functions that we don't have time to talk about today. So we've talked about the nervous system from at the level of cells, at the level of tissue, and now let's have a look at it at the gross level, the naked eye level. And we can see essentially in this very nice diagram of a dissection the two broad parts of the nervous system. The brain and the spinal cord make up the so-called central nervous system, which essentially is where computations take place, where memories are formed, etc. However, the central nervous system needs to communicate with the rest of the body, and that is through the so-called peripheral nervous system. The peripheral nervous system we can see in this diagram starting to form as all of these nerves emerging from the spinal cord, which ultimately get distributed to the skin and the muscles of the limbs and other parts of the body. So the central nervous system and the peripheral nervous system are the two broad categories of nervous system within the human body. So how can we learn about the nervous system, about its structure and function? Well, the oldest technique that's been used, and this is a technique that's been used for thousands of years, is the technique of dissection. So you can remove part of the nervous system and you can look at it. You can cut it up. You can look inside of it. So, for example, here is a sheep brain that's being dissected, and the person here has cut it down the midline and is looking into the various cavities within the brain. Here's a similar view of the human brain. So you can cut up the nervous system, you can dissect it, and you can get a lot of structural knowledge by doing that, looking at how the thing is put together. But actually, you can't get all that much function from this. As you can imagine, how are we to infer by looking at the cerebellum and how it's all folded that has some role in coordination? We can't really get function from structure at the level of dissection. Some people did, though. In the 19th century, there was a large group of individuals who called themselves the phrenologists. And phrenology was the study of lumps on the head. So the phrenologists believed that various regions of the head, the size of a lump on that region, correlated with a person's tendency to particular characteristics. So, for example, this region just above the ear was correlated with caution. So they believed that if there was a larger lump there, the person would be more cautious, for example. Now, to our modern point of view, phrenology seems crazy, ridiculous, a pseudoscience, and that's indeed what it is. However, the phrenologists did have some inkling of something going on which we are now aware of. Because what we now know is that all these different regions of the brain do indeed have different functions. So there is localization of function within the cerebral cortex. So the phrenologists thought that, it's just that they had the slightly misguided idea that you could feel those things through the skull and the scalp. Now, you can only get so far with dissection, as we said. And if you want to study the function particularly of the human nervous system, one large area that has been used over the years is natural experiments. Now, natural experiments, it's a bit of a euphemism. Really what it is it's diseases, it's tragedies that have befallen people. And neuroscientists historically have looked at these individuals and tried to correlate the damage that was done to their nervous system with the function that was lost. So a classic example of a natural experiment, for example, is the guy on the right here. Most of you would recognize his face from a large national campaign that's still ongoing, because this guy has had a stroke. So you can see that his mouth is drooping on this side, his eyes drooping a little bit there. Okay, he's had a stroke. And what's been done over the centuries is that neurologists have looked at patients who have suffered from strokes, looked after them during life, and then when they died, they took their brains out and looked at where the damage had happened. So, for example, in this guy, we can see his face is drooping here, so we would predict that we should find damage in the right side of his brain, since the left side of his face has been damaged. Okay, so that's what's been done. Look at the symptoms a patient has, look at their brain after they've died, and try and link the two together. Another classic example of this involved this guy here. Now, this chap was called Phineas Gage, and Phineas Gage had a very severe accident. Essentially, this long iron rod, which he used to squash gunpowder into holes that he drilled in boulders in order to blow them up. This long iron rod shot out of the hole and went under his cheekbone on the left-hand side, straight out the top of his head. And in the process, it destroyed much of the frontal lobes of his brain. Now, this proved very, very interesting for many generations of neuroscientists, because Phineas Gage's personality changed fundamentally. In a nutshell, he went from being quite a decent chap to being quite a bad guy with a fundamentally changed personality. So this told us something about what the frontal lobes do. Now, the problem with natural experiments is that you're relying on accidents, and accidents by their very nature are random. So the location of strokes, you can't predict, you can't control where a stroke is going to happen. So if you want to look at the function of very, very specific areas of the nervous system, you have to rely upon animal models. And this is a huge area which has been used in the elucidation of the function of the nervous system, the use of animal models. So basically what you can do is you can take an experimental animal, in this case using a rat, and you can make a small hole in its head, and you can either stimulate or destroy a part of its nervous system. Now, of course, as we know, rats can't talk to us. They can't tell us about how they feel. So in order for us to work out the often very subtle effects of these lesions, what we have to do is measure the effect on behavior. So in this case here, this very complicated apparatus has been set up in order to measure, for example, how frequently this rat presses a lever or some other aspect of its behavior. Okay, so using lesions in animals, what you can do is damage or stimulate one small area of the animal's brain and then measure a change in its behavior. And this has been done, and this has helped us to treat a whole range of diseases. For example, modern treatments for Parkinson's disease would simply not have been possible without work like this using experimental animals. So that's focusing on function. But what about the wires themselves? So how might we trace the connections within the nervous system? How might we trace, for example, the trajectory that axons take in going from the brain to the spinal cord? Well, here there's a lot of methods which are available to us. In the top image, this is actually some work which I did when I was doing research, showing the trajectory of axons in the nervous system in the sensory part of the nervous system. So in this experiment, what I did was injected a virus containing a fluorescent protein, the gene for a fluorescent protein, into sensory neurons. And so any sensory neuron was labeled with this green protein. So we could see here, for example, in this part of the spinal cord, we can see where all these green axons are ascending towards the brain. So we can inject viruses containing fluorescent proteins into neurons in order to trace where they're going in the nervous system. Alternatively, we can inject dyes, simple fluorescent dyes. So the bottom image, this, believe it or not, is the tongue. And what's been done is the sensory neurons supplying the tongue have been injected with a fluorescent dye. And you can see that they have filled up the sensory projections coming from the individual taste buds of the tongue on both sides. So very nicely, by injecting dyes, we can see where the axons of neurons terminate within the body. Now, the examples I've given before are only used to study the paths of individual neurons. Those dyes or those viruses I talked about do not cross synapses. However, there is one particular virus which is in fact very good at crossing synapses. And that is the virus which has infected this dog here on the left, namely the rabies virus. Now, rabies essentially gets into the body. So say if this dog bit you, its saliva is full of rabies viruses, and the viruses would get into your skin. They would then infect your sensory neurons, and then the virus would travel up towards your brain by crossing the various synapses in the sensory system until it ultimately overwhelmed your nervous system and caused you to undergo some behavioral changes, as have happened in this dog. So rabies virus is able to cross synapses. Now we can exploit that fact, because what we can do is, using genetic engineering, we can remove the pathological genes from the rabies virus, and we can replace them with genes for fluorescent proteins, meaning that we've now got a virus which can make neurons light up, but which can also cross synapses. And that's been done in this experiment here shown on the right. There's a lot of detail, don't worry about the detail, but essentially what was done is a muscle, one single muscle was injected with a fluorescent rabies virus. And what these scientists were able to do was to observe the propagation of that virus up towards the motor area of the brain that supplied that particular muscle. So each of these dots is a motor neuron that supplies that specific muscle. Okay. Now, what else can we do? Well, as I alluded to earlier, you can stimulate parts of the brain. So here is a cross-section of rat brain, and you can put electrodes into the rat brain and stimulate and see the effect on behavior, as we've talked about. But of course, that's not very ethical in humans. However, in humans, what we can do is we can use magnetic stimulation to stimulate or even to switch off different areas of the brain. And this is a technique called transcranial magnetic stimulation, or TMS. And what you can do is apply a very strong magnetic field to a region of the brain. And as you might know, electricity and magnetism are two sides of the same coin. So by supplying this strong magnetic field, we can cause electrical activity in that part of the brain, i.e. we can stimulate it, and then we can see the effect on the behavior in the human body. Most of you will have been familiar with images like this. So this is an example of functional magnetic resonance imaging, essentially looking at changes in the blood supply to different regions of the brain when the brain is doing a particular task. The colours here are in fact irrelevant, really, but what we can see is that there was increased activity in the visual part of the brain at the back, as well as in the auditory regions of the brain at the sides. So this is an fMRI scan of someone who was watching TV, right? So they were looking and they were listening using their visual and auditory cortex. So we can use functional magnetic resonance imaging to correlate a task with activity in different regions of the brain. I'm not going to linger on this, but we can use that on all parts of the central nervous system. And in fact, what this is showing is it's showing the propagation of pain signals up the spinal cord, ultimately to the brain. So we can even use this on the spinal cord. Another imaging method derived from MRI scans is known as diffusion tensor imaging. And these images have also made it into the popular psyche because they're just amazingly beautiful. What DTI, or diffusion tensor imaging, does is it looks essentially at the movement of water by capillary action through axon pathways. So if you imagine thousands of axons all running in the same direction, water moves along that by capillary action, and the MRI scanner can pick that up. And beautifully, we can see then on these scans the trajectories of the various axons. So we can see, for example, axons crossing the midline here in the corpus callosum, connecting the two hemispheres. We can see the motor pathways going down towards the spinal cord. And this is becoming a really powerful technique to chart the course of axons through the brain and spinal cord. Now, what we've talked about so far has really been primarily about structure. There's been some allusion to function, but I want to be a little bit more functional now. And I want to talk about a technique which is really not bang up to date, but within the last 5 to 10 years has really taken precedence in the neuroscience field. And this is known as the technique of optogenetics. Before we talk about optogenetics, let's take a step back and let me explain to you why I'm showing you a picture of Jennifer Aniston. Now, there's an academic here at Leicester University who earlier on in this century did a number of interesting studies using patients who were undergoing surgery for epilepsy. So these were patients who the surgeons weren't sure where the area of brain that was causing their seizures was. So what they did is they put electrodes into these patients' brains in order to catch a seizure in action, so to speak, to exactly locate where it was so that they could then cut that bit of brain out. But what this guy did was he came along and he recorded from these electrodes while the patients were not having seizures. And he recorded from these electrodes whilst he was giving these patients a task. And the beauty of these electrodes that were being used is that they could actually focus on individual neurons. Using some clever mathematics, they could just filter out the signal from a single neuron. So he was recording from one neuron, and what he did is he showed these individuals hundreds and hundreds of pictures of people. So he showed them hundreds of pictures of individuals, okay? Some of them were famous, some of them weren't. They'd shown hundreds of pictures and still the neuron that he was recording from was silent. Until he showed this individual a picture of Jennifer Aniston, at which point the neuron lit up. He thought, that's interesting. So he found another picture of Jennifer Aniston, and lo and behold, the same neuron activated again. Okay, that's interesting. So then he thought, well, in that case, let me write down Jennifer Aniston's name, and I'll show that to the patient. He showed them the written Jennifer Aniston, the neuron lit up. Okay, what about if I talk about her boyfriend at the time, who I think was Brad Pitt? He talked about Brad Pitt, and the Jennifer Aniston neuron lit up. So he had discovered what became known as the Jennifer Aniston neuron, which caused quite a stir in the neuroscience community, because this neuron had actually been proposed by philosophers centuries before, but it was called the grandmother neuron, which was the neuron that was meant to be activated solely by when you were thinking about your grandmother. Now, further work has confirmed that the Jennifer Aniston neuron doesn't really exist. It's actually one of hundreds of neurons forming a network or an engram which encodes the concept of Jennifer Aniston. These are now called concept cells. So these neurons form networks which represent a concept. That could be Jennifer Aniston, it could be the concept of a book or a light switch. So that's very interesting, shown in humans, but can this be confirmed in animals? Well, this has been confirmed in animals. And that's why we're showing this picture of a rat here with a little light on its head. So what was done in these animals was two things. First of all, the neurons were genetically engineered so that if they were activated, they expressed a colour. All right. So the animal was given a task to do, which meant that a particular population of neurons were activated intensely. Those neurons lit up. So this pattern here of red dots represents the memory engram of this task that the rat was taught. Now, secondly, not only do these neurons light up, but they themselves become responsive to light. And in fact, the task that was taught to the rat was an association between a stimulus which caused a fear response. Okay, so it was classic Pavlovian conditioning, if you like. So this is the engram that encoded the association between a certain stimulus and a fear response. But what the researchers then went on to find was if they specifically lit up these neurons, they got the fear response from the animal in the absence of the stimulus. So this was believed to be the first demonstration of the cellular substrate of memory, okay? All related to the idea of our Jennifer Aniston neuron. Now we're coming to the end now, and I just wanted to talk to you in the last few minutes about a cutting-edge technique which is known as connectomics. And some people believe that connectomics really holds incredible promise for the future of the human species, effectively. So basically, the aspiration of people who are interested in connectomics is to map every single connection within the human nervous system. Okay? So they want to know the location and the functional properties of every single synapse in the human nervous system. And the way that they intend to do this is to cut the nervous system up into very, very thin slices and examine each slice under an electron microscope. So what you can do is you can take a cube of tissue here and you can slice it into many, many thousands of slices, looking at each with an electron microscope. And you can look at a particular neuron, particular dendrite or axon, and you can reconstruct that in 3 dimensions. And you can do that for all of its neighbours as well, until you've derived this incredible detailed map of the connections. This image on the top left is in fact a connectome of a very small region of the retina in the eye. So the idea is, let's map every single one of the connections in the nervous system of the human. But there's a little bit of a problem here. Now, this has been done already in an invertebrate known as C. elegans, which is a small roundworm, about 1 millimetre long. The nice thing about C. elegans is that we know about all of its cells, and we know that it has precisely 302 neurons. This on the top right is the connectome of C. elegans with its 302 neurons. So this is the map of all of the connections within its nervous system. This connectome took 12 years to derive. So it took 12 years to take a 1 millimetre long worm and to map all of the connections between its 302 neurons. Now, the human nervous system, the human brain, has approximately 100 billion neurons. For those of you who are mathematically inclined, I invite you to do a simple calculation to work out the scale of this problem. It is massive, and even though in principle we can do it, we don't currently have the technology that's going to enable us to get an answer to the human connectome anytime soon. But if we get this connectome, what would we do with it? Well, that's a very good question. Some people believe that once we know every single connection in the human brain, well, shouldn't we then be able to simulate that brain, maybe in a computer? Could we maybe get someone's connectome after they died and reincarnate them in silico? All of these things might sound like something from a Black Mirror episode, and in fact they are, but we need to start thinking about the implications of technologies like this, because they could actually be upon us before we're ready to deal with the moral and ethical implications that they might have. So that's all I've got to say to you. Thanks ever so much for listening. And I wish you all the best in your future studies and applications to medical school. Okay, goodbye.